Presentation is about gene cloning by Zayden and me, Santiago. So what is gene cloning? We're gonna explain it really simply because it's a really complicated topic, fairly new. So one of the first steps in gene cloning is you have to isolate the gene. So you look at an organism's DNA, it could be a plant, it could be an animal, anything like that that's living, and you try to find the gene that you want to copy or clone, and then you try to isolate it by extracting it.
So they will extract a gene from an animal or a human that they like, and they will place it in an organism to see how it turns out. They place it into the organism to see how mutations work and if it's safe to be used.
So here, it's not like you have to clone it every time, one cell at a time. Cells in general already multiply by mitosis and meiosis. So people just you put them in an environment where they're allowed to do that, so you don't have to clone a single cell so many times. So it it lets it do it by themselves. And as the whole cell reproduces, it copies so many times basically. It's similar information. It just it copies really quickly because it's they make an environment so it can specifically its main purpose is to multiply.
So key players in gene cloning is a targeted gene. So they want a specific gene and they'll extract it, and then the vector, the gene DNA is often carried in a plasmid. A plasmid is a small piece of DNA. It's circular, and they'll put it in other vectors. Key players in gene cloning, typically fast-growing bacteria, yeast is easy to multiply. So the genes will break off, and more will get made. Enzymes, specialized proteins that is cut and join DNA molecules precisely. So it's pretty much building a whole new organism.
Yeah. So there's the actual steps, and then because it's a fairly new topic, there's still research that needs to be done. So there's some conceptual steps. So it's similar to the first slide, gene isolation. So now that you actually have the thing and they're multiplied, you have to pick out a gene that's not modified. It's a good gene, a good one that would actually be the right fit for what you're trying to insert it into. So and then the second step, you would insert it into the vector. So what is a vector? Vector is the cell that you put the DNA into. It's kind of like a capsule. That's what you a vessel.
Transfer into the whole cell. Introduce the vector, the carrying gene to the whole cell. So let's take that DNA they took and they'll put it into the vector. Replication and expression. So the whole cell will copy it and they'll multiply more and more, and then they'll extract the copied one to see if it's exact as the original one.
So selection, you have to identify and see which one would actually work, and then after you let it multiply in the organism, you have to look again to see if it was the right fit, to see if it's actually doing what you wanted it to do, just to see if it was a successful clone. And there's a lot of applications to this because it kind of sounds like magic and stuff, but it's actual day-to-day usage for these things. You could do cloning for an organism that has more insulin than a human, and you could insert it into someone that has something like diabetes, and this could help them not get rid of the disease, but it would help manage it way easier. You could get growth hormones, more natural vaccines that don't really have to be applied every so often, because you would have the cell multiply by itself inside the body, and you could also cure some genetic diseases that could be fixed.
Diverse applications of gene cloning. So the research for it is they have to study the gene function. They're going to extract that gene and they're going to study it and how it helps understand the mutations. So there always are mutations when trying this, and they have to understand the mutations to prevent them from happening and research them. Advanced gene and molecular biology. So they just pretty much try to research about everything before actually proceeding with the experiment. Diverse application of gene cloning. Agriculture, for an example. So these are for crops like GMOs, and so they can reproduce stuff that can handle pesticides and the bugs around them, so they can build crops quicker. Instead of if there's a drought, they can handle all the harsh climates.
Yeah, so some more diverse applications of gene cloning would be like not even because he said crops. Like you
Yeah.
could make plants that have a built-in resistance to like pests and stuff. But you could also do like detergents and biofuels. So instead of like with biofuels, you have to use like compost and stuff. So you could like multiply like fuel for that and not have to use existing fuel. And some more advantages of gene cloning could be large-scale production of like useful proteins like insulin. If you can make that like available like worldwide easily, then that would be like really helpful towards a lot of people. And it would just be like a overall like a great advancement for medicine if it was like the technology was more like readily available for everyone and it was more like developed.
Advantages of gene cloning, it helps scientists understand the function of genes and their mechanism more, and then it helps target traditional breeding methods. So we could be cloning people instead of having to have the traditional methods of having people have sex. Ethical and safety concerns. So gene cloning, especially gene modification, raises a lot of questions. If it's safe, the mutations, like some mutations can have, like you say, you could have an extra ear, and you don't want that to happen. So there are a lot of risks to it. Strict regulations, ethical guidelines on cloning, research ensure careful and responsible use. So it's really precise, and you have to do a lot of research about it.
So that's what we're saying. And thank you for listening.
Are there any questions about gene cloning per se?
Oh, so long.